transcription have a really high mutation rate, or does HIV just have an especially crappy reverse transcriptase?
Yeah, no, most viruses, retroviruses in general, reverse transcriptase doesn't have what we call a proofreading function. So when a mistake gets incorporated, there's no way to take that mistake out and repair it. So it's in contrast to a virus like influenza. Now, influenza it's a DNA polymerase which helps it replicate. That DNA polymerase is much less error prone, so there's a lot fewer mutations that get incorporated. So it's a little bit easier for us to make a vaccine against influenza. Now it's not easy to make an influenza vaccine, but we can look at strains that are out there and predict whether or not they will emerge, and we can make a vaccine towards those strains, and we know that when they do emerge, they won't change that much. HIV, you're producing every day. When you're infected with HIV, you're 10 billion viral particles, every single day. And you're seeing, your body's seeing, HIV, the length of the genome is only 10,000 bases, and every single base can potentially be mutated. So you're seeing every single possible variant of HIV almost on a daily basis. I mean, it's unheard of. I mean, it's just the mutability and the ability. That's why I titled this lecture, co-evolving epidemic, because this virus continues to evolve. I mean, we throw drugs at it. It continues to evolve as it moves into different populations. I mean, the strain that infects Chinese is different than the strain that infects Europeans. That's slightly different than the viruses infecting Africans. So the virus evolves. Now, our own immune system, though, continues to evolve, and we continue to evolve and think about better drugs. So it really is this co-evolutionary struggle, and so far the virus has been winning. But why is it so difficult? I mean, for one, when you're talking about a sterilizing vaccine, you're really talking about making antibodies that recognize the viral envelope and just prevent it from getting into the cell. And this viral envelope is, like the rest of the virus, it's just very, very mutable. It can tolerate lots of different changes in its structure. So you'll produce an antibody one day that recognizes the virus, and then a week later the virus has evolved and the antibody no longer recognizes it. But it's also, if you notice this envelope, it's a trimer. It's actually three individual envelopes sort of coming together. And what HIV does is, some of its most vulnerable parts of that envelope, it hides it in the sort of the center of that trimer. So only when it's going to bind to its target cell does that trimer open up and allow the virus to come in. So it does a great job at really hiding these vulnerabilities and the stuff. So the antibodies can't recognize some of these really vulnerable epitopes that are hidden there. Again, what I was just saying about, though, we really don't know. Like, when HIV is spreading from one human being to the next, we don't know if it's spreading as a virus outside of the cell. Like, a cell of person A produces the virus, and then that virus is just kind of floating out in the middle of nowhere and then gets to person B, or if it's actually, the virus is inside of the cell. And then when I pass it on to you, I pass the virus on inside the cell, and then from inside that cell it goes to another cell. And this makes a big difference for making a vaccine, because if HIV is out there and exposed, it's going to be much easier for the immune system to see it. If HIV is kind of hiding in cells, and then when those cells are in semen or whatever, when they then move into the next person, it's very difficult for the immune system to see that. So, okay, just very quickly here, I'll talk a little bit about the kind of stuff that we do at the Gladstone. Okay, that's where, we're actually down there. Okay, the whole floor. Let's see here. The Grant Lab. The Grant Lab is a very international lab. They work, one of their main sites is down in Peru, and they're testing pre-exposure prophylaxis lacks this. So the idea here is to give people drugs before they get infected and see if the person who's taking antiretrovirals on a regular basis, does that help protect them, and does that decrease the transmission of HIV. The burden lab. So Eric's lab really works on HIV latency. You know, as I told you, HIV can infect some cells and it just kind of hides out in those cells. It doesn't move around. And so what they believe, you know, this makes it impossible to cure HIV. Right? You know, if you go on antiretroviral therapy, you've got to stay on it the rest of your life, because there's always the possibility, even if you drive your viral load way, way down, there's always that possibility that HIV is going to pop out of some of those latent cells and then begin to, you know, infect you. So what they do is they try to discover drugs. They take these cells that are latently infected and they do a large-scale screen to try to find small molecules that activate and force HIV to pop out. And then once HIV pops out, it should then be susceptible to, you know, reverse transcriptase inhibitors or protease inhibitors or whatever you're going to treat it with. The OT lab. Melanie, she's a fabulous scientist. Her work is really aimed at trying to understand HIV once it integrates into the genome, how it is expressed and how it starts to produce its proteins. And so she looks for host proteins. I mean, these are, you know, human proteins that the virus has to co-opt and use in order to get it to you know, transcribe and produce its own viral protein. I mean, HIV is really, it's a tiny virus. HIV is like I said, it's a 10,000 base pair fragment RNA that encodes 14 proteins. You compare this to something like a herpes virus. A herpes virus has 300 proteins. So HIV is just this tiny little fragment of RNA. So it's got to really make use of our human proteins. And there's a big effort underway to understand what human proteins HIV needs. Because the hope is that you might be able to find a human protein that HIV needs, and you can target that human protein with a drug. And that human protein isn't going to be able to mutate. And so then once you take that human protein out of the picture, HIV can't use it, and the viral life cycle stops. And then this is the green lab, which I'm in, and one of Warner Green's main avenues of research is to look at a protein called ApoBec3G. This is an antiviral protein. All of us produce this protein, and what it does is ApoBec hypermutates the HIV virus. So it's ApoBec that's incorporated into viruses, and then when the virus goes to infect the target cell, when the virus starts undergoing reverse transcription, that's when ApoBec intercedes and basically mutates the virus so much that the virus can't produce any properly folded proteins, and the whole life cycle stops. Now, the downside to all of this is that HIV produces its own protein, which recognizes ApoBec and targets ApoBec for degradation. So this is how HIV gets around the block from ApoBec, by causing the destruction. So what our lab is doing is we're looking for small molecules and drugs that will block this interaction between the HIV VIF and the human ApoBec. So that way it can keep this in the cell and it can, you know, continue its antiviral activity.